So thank you, everyone, for contributing to this session. This is a first kind of meet the board science session. So they're not scary. They're just human beings. They will ask you nice questions, and they'll be really interested to hear your science. That's to the students and postdocs. To the board members, they're not scary. They're humans. They're just interested to hear what your questions might be and talk about their science. So this is the first of hopefully several of these. So today we'll focus more on the early careers researchers and the science and the individual scientific projects. And Carl Logan is going to compare
all drug targets and potentially curative targets. So eczema is very common. People here will know about it, and it has a major impact on health. And I do dermatology, so we see patients with more severe disease needing therapies that modulate the immune system. And so these therapies have side effects, and they have an incomplete response rate. So what patients often ask is, which treatment is best for me? And that's quite hard, as we don't know which patients will respond to which drugs, so we often cycle between them. And then at follow-up appointments, it will depend on how patients have responded. So if some people don't respond at all, and they ask what else can be tried, and apart from those treatments, there are no other effective long-term treatment options. Some people do really well on these systemic treatments, and the skin looks normal, but as soon as you stop the drug, the eczema comes back. And so they sort of ask you what's the cure. Otherwise, you need to continue long-term, and that's expensive. So some of these drugs, like dupilumab, cost around £10,000 per year. It also exposes patients to side effects, and the long-term safety isn't known for the new drugs. So my project is BEACONomics, which aims to answer these questions, and I'm going to give an outline of the project plan today, where we aim to describe the cellular and molecular landscape in eczema, try to predict which patients will respond to which therapies, and also more of a generative AI context of creating models for cell modeling with the ultimate impact of healing. So it's quite common to do skin biopsies from skin for diagnosis. Sometimes in eczema, you're not entirely sure if it's something else. And this is what skin looks like under the microscope. So the top layer is the epidermis, which sort of comes off if you have an abrasion or scratch, and then you get an idea of what the skin underneath looks like. But this is quite a crude description of eczema. And now we can do much higher resolution profiling. So taking all the cells in the tissue and measuring sort of 20,000 and upwards genes per cell. So it's a really detailed representation. I think that's important for describing any system, even complex systems. So with the weather, if you look outside, you can predict what the weather will be like the next day. But actually, if you have humidity, pressure, temperature, and all these other predictors, you have a much better understanding of the system and short-term prediction for that. And what we can also do is measure in the actual tissue where thousands of genes are expressed. And so this allows us to link the tissue that we see, that we use in clinical diagnosis quite a lot, with this really high resolution gene profiles that we generate. So how can we use these high resolution profiles to have a clinical impact? So there are two unique parts of BEACONomics, which I'm excited about. The first is that it's in collaboration with a clinical trial, so the largest platform trial in eczema, recruiting more than 500 patients nationally to see which one of these drugs that we commonly use is most effective. And that also gives us extra data like treatment response in previous treatments. And we take that further by an open targets collaboration where we perform this multimodal profiling that I just described, and we perform it at scale, which I'll describe more in the next slide. So the largest sort of single-cell study in eczema is very new, a preprint from the Brood, but they only did sort of 30 samples at one time point. Whereas here we're doing 50 patients at 4 time points before and after treatment. And we're also doing blood and skin tissue. We're doing whole genome sequencing plus spatial plus single cell. But I'm going to focus on the skin and the single cell and spatial. So by taking these high modality profiles, we can actually look how the skin changes over time at a very high resolution. And drugs like JAK inhibitors and methotrexate we use in diseases like inflammatory bowel disease and rheumatoid arthritis, where you can't take tissue biopsies at the same scale or even in the same time point. So this is a really unique view into human inflammation in vivo and how patients respond to different drugs. And also by looking at, say, non-responders, we can see how they're different to responders and potentially identify drug targets. And in the people with clear skin at day 90, we can see how their skin is different to diseased skin, and then potentially identify how this memory in the skin is mediated. And then because we have the trial metadata, which is rare for this type of study, we can start stratifying patients into patients who we think will respond and not respond. And one potential use of this, which I'm just going to describe, is using that spatial transcriptomics data I mentioned before, where we have this really large-scale samples of tissue and the gene expression. So we can think about, for each tissue here, we can break it down into really small regions. In each region, we'll have a label. Is it from a responder or a non-responder? And you'll have a really large amount of data here. And so then by training a machine learning model, you can identify which areas in that tissue are most predictive of, say, non-response. So the areas that are found in non-responders and responders won't give you very helpful predictions, but actually, the areas specific to non-response, you'll get high predictions that this slide, or like in machine learning would be the bag, belongs to that label. And so then we can see in tissue which areas are most associated with non-response. And we have this spatial H&E quite commonly available clinically, so then it helps sort of inform our decisions in clinic. And then I also want to talk about generate. So I mentioned how we could describe in detail skin compared to how we could describe it in the past. And with that scale of data and detail, then we can start thinking about sort of generating cells. So we know generative AI is very powerful. I guess ChatGPT-4 is the most commonly known example of this. And the slide has moved a bit, but you can guess what the next word is going to be here. The mouse ate the, it's probably cheese in this context. And so ChatGPT works by this sort of autoregressive prediction of the next token. So then you make sentences by predicting the next word, which sounds very simple, but that's the whole sort of emergence property that makes it exciting. And so just as we can think of sentences being made out of words put together progressively, we can think about how we can make cells by progressively putting genes together if they're ranked. So if you see these genes, if you do like skin work, you have an idea of what the next gene is going to be. And so you can continue this on and generate cells, which might sound very ambitious because there's trillions of cells in the human body. But the flagship for AI in biology is AlphaFold, where we see like very powerful and useful predictions in biology. And I just wanted to emphasize that an important part of that was generating the data to train the models. So this started in the 1970s with the protein data bank, which was open access, and progressively more and more data became available, such that AlphaFold could be trained. And there wasn't anything hugely new in the sort of architecture itself. It was this commitment to open access by scale data. And so that's promising for single cell because of the Human Cell Atlas, which is a global initiative aimed at generating, promoting profiling of cells at a really large scale. So there's already more than 30 million cells available, and that's a really high detailed description of all the genes that they're expressing. And there's more than 1 million skin cells available too, and more than 2 million if you include disease. So that's exciting because it gives us a promise that we can train similar models for predicting cells. And how is that useful? It's quite hard to explain, but one idea is that you have this really understand the representation of a cell. And then with our metadata that sort of has information attached, whether they responded, what drug they had, then you can start to predict say, how patients would responded if you gave a different treatment. So what would happen if we change the treatment? This is a bit of a simplification, but because we're combining a text prompt here with a cell generation, but I guess we saw that with DALL-E or Midjourney, like these text-to-image generators. So it's definitely possible using those sort of architectures. And actually, it is difficult to understand because it is difficult to explain why ChatGPT is very useful, even if it's predicting the next token, and that's how it learns. But it actually has more complex behavior than that, so sort of this immersion theory. So I guess that's why the whole field is quite exciting. And so then how do we actually have a clinical impact? Well, we're profiling non-responders and excellent responders to see what we can actually target to offer these patients new therapies in clinic. We're beginning to have a more precision medicine approach where we make more informed decisions about which drug we start. And then also I mentioned about in the future using generative models to predict how cells and how patients will respond differently to different treatments. And I wanted to finish on this slide. So at the Sanger, we can do really unique science, and I'm really excited about the project I'm doing. It's sort of not available anywhere else in the world. And from my clinical practice in dermatology, I've seen lots of patients with very severe eczema that also is very questions. And actually having 5 skin biopsies, which they have in this trial, is a major undertaking. But patients are sort of very committed to contributing to research. So it's nice to operate in both worlds and make sure that our research is clinically informed, but then also in the clinical world that we use what we find in research. And I think in healthcare, we'll definitely get more and more large-scale data like transcriptomics and genomics over time. And it's important to use that for patient benefit. And that was everything I was going to say. This slide would be a whole other 10-minute talk. Like everyone's very helpful. It's a really good environment to work. The slides didn't work properly, but yeah, thank you for listening. Yeah, it's a good question because eczema is thought to be a combination of the disrupted barrier, like a leaky barrier, plus the immune system being a bit weird. And you get monogenic disorders from each of those that can cause it itself. I would say you see them in both. You see like the type 2 cells, and you also see disease keratinocytes that are proliferating faster that you see in clinical practice, right? Like the skin scaling and peeling off. But from single cell, we also see fibroblasts as being abnormal. And now they probably bring the T cells in and keep them there. And that's sort of new from single cell. Before we saw they were all the same. Thank you.
Had you looked at, or are you looking at adjacent normal or even control tissue in relation to exposure?
Yeah, probably in 2 ways. So at baseline, they have a non-lesional biopsy. So from skin that looks normal, but is in someone with eczema. And then also as an external reference, we're making like an atlas of the skin. So that more than 1 million skin cells is what we're assembling. And so that's our sort of healthy control.
And are you just using white skin or are you using different ethnicities?
So in the trial, there's no particular drive for that. Like the major recruiting center is in London, in King's College, and they're expecting like diverse tissue. Like my clinical practice is actually in Royal London, so East London, where actually we see mostly patients with mostly Bangladeshi patients with eczema, and they have a slightly different phenotype. So hopefully we do capture that. We're expecting to too. Thank you.
So I am Cecilia from the branch out in Parasites and Microbes, and I'll take us on a little tour about bacteria and pathogens in the lung during chronic lung disease, which is some work I'm looking to do for my PhD. So the lung microbiome in health is a very low microbial load, and this is completely opposite to that of the gut. And this microbial load is kept really low by the constant clearing by the immune system and mechanical actions such as coughing. So in a healthy person, we have very low microbial load and a clearance system that works. In chronic lung disease, these clearance systems are disrupted, and we see lots of mucus accumulation, which favors local microbial replication and high microbial loads. And these high microbial loads then drive constant inflammation and lung damage. And a very good example of one of these chronic lung diseases, I'm sure that you've probably all heard about, is cystic fibrosis, where the lungs are characterized by a very high pathogen load of specific pathogens, of non-pathogens that drive constant inflammation and resulting in extremely destructive lung damage. So cystic fibrosis is not caused by microbes or bacteria. It's a genetic disease caused by a mutation in a transmembrane regulator, but a lot of the disease progression is driven by these high microbial loads. So from studying cystic fibrosis, we have identified the key pathogens that drive this destructive cycle and the high microbial loads. And we have also appreciated that during chronic infections, that the pathogens will diversify and adapt to this environment in the lung. And on here is I'm showing a constructed history of one of the common pathogens, Mycobacterium abscessus, where we see adaptation in the key virulence genes. Hence, the bacteria is able to survive in the lung environments. 99% of what we know about chronic infections comes from sputum. And this is because it's the most readily accessible sample type. But then we also know that the lung is a very complex organ with lots of structures and lots of compartments. And what we do not know is how well sputum represents this structuring and compartmentalization in the lung. We also know that parallel evolution happens during chronic lung disease, and essentially, and on here we have identified parallel evolution in key virulence systems within patients with a chronic mycobacterial infection, and this can only be explained by spatial isolation or compartmentalization of bacterial populations in the lung. So from our current understanding of within host evolution, we know that space is important. But what we do not know is in these spaces where the bacterial populations are living in the lung, do we have co-infecting bacteria in those spaces? And how do pathogens behave in different cellular contexts? So to understand this, we can look at pathogen distributions across the lung, and we can sample differently. And we can sample either using bibronchoscopy or explanted lung tissue. And I will share some work that we have done on explanted lung tissue from different parts of the lung. So we obtained some explanted lung tissue from a cystic fibrosis patient who had a chronic mycobacterial infection. And we noted that the spatial distribution of this pathogen in the lung correlated very well with the radiology images that were obtained just before transplantation. So this spatial distribution here correlates very well with this region here on the radiology. And then we also noticed that there was diverse subclones in the lung tissue. And what was most striking was we identified subclones that had not been detected in sputum that was collected during transplantation. So essentially, we're getting more subclone resolution when we look at the lung tissue as opposed to the sputum. We also identified co-infecting bacteria that were co-located with the mycobacterium in the lung tissue. And we thought, well, these results are very encouraging and very interesting. How about we take a holistic approach, and instead of just looking at one pathogen, we look at more than one pathogen, and also at the same time look at how the host is responding to the presence of these pathogens with the lung tissue. So we hypothesized that by overlaying the gene expression information from the pathogen and the host, which is the human lung tissue, and the lung histology, we will be able to co-locate where these microbes and the different cell types are in the lung. So in collaboration with Duke Hospital, we obtained explanted lung tissue from a cystic fibrosis patient who had a chronic mycobacterial infection. And we got some very interesting data. We were excited and very thrilled that we were able to detect bacterial reads from this lung tissue block. And what was quite striking was we identified the top known species in CF, so Mycobacterium, Klebsiella, Staphylococcus, and Pseudomonas. And what stood out was the different distributions across of these key pathogens across the different parts of the lung. We see in the left lung is mostly dominated by Staphylococcus aureus, and the right lung is dominated by Klebsiella pneumoniae. And we thought this Klebsiella was very fascinating because Klebsiella is an opportunistic pathogen, and it is known to colonize the oral cavity and the GI tract. So we think the presence of Klebsiella in the lung at this point was due to contamination during pulmonary exacerbation. So a lot of coughing, you get GI reflux, and you get some Klebsiella in the lung. We went on to do some histopathology on the tissue, and we noted mucus-filled airways, which is a very well-known characteristic in cystic fibrosis. And very interestingly and strikingly, we identify lots of granulomas at different stages of formation. And granulomas are essentially structures that form during chronic infection as the immune system is trying to wall off a foreign body or a bacteria. So bulk RNA-seq tells us there is bacteria in the lung, and the granuloma from the histopathology and these features are telling us there is inflammation. What we do not know is where is the bacteria in this structure and in what cell types especially. So to answer that question, we are adapting spatial transcriptomic methods by adding bacterial probes. And before this, spatial transcriptomics has largely been used to profile gene expression in host tissue. So by overlaying the transcriptomic information of both the bacteria and the host tissue and the lung histology, we will be able to collocate pathogen and host cell type. We will extend this to a tissue collection of 12 CF patients who had a chronic Pseudomonas infection at time of transplantation. And then we will extend this to a COPD cohort where we have punch biopsies and from patients at different stages of disease. So microbes have been indicated as drivers of disease progression in COPD, and it would be really nice to understand the distribution of these microbes in the lung. Ultimately, by understanding how microbes are distributed in the lung and how they interact with the host tissue, we will be able to inform better clinical interventions and hopefully identify novel targets for therapeutics. This work would not be possible without the great guidance and supervision from my supervisor, Josie Bryant. It is a great collaborative effort between pathogens and cellular genetics, the Tekman Lab and the Bayrector Lab, and departments across the University of Cambridge, Palm, and Celgene. Thank you so much for listening.
Thanks, Cecilia. That's a great talk. Are there any questions? Mark, I think.
How would you go about teasing out causal inference here? You're going to find lots of things grouping together, and you won't know what the chicken and the egg is. How are you going to disentangle that?
Good question. I don't have an answer at the moment. Yeah. I guess we'll have to get us together with the Celgene team to know which can pass or.
Are there any other questions? Yeah, Dan.
DD patients, because they're at risk of again, winter residual viral infection that's obviously a change. You protocols the word. How are you kind of controlling that? So for those of you who want to see the impact of the microbes on.
So for the COPD cohorts, I think we only have biopsies from early stage and late stage of disease. And our control would be a healthy population. So we don't have, I think the ideal would be to have longitudinal sampling from the COPD cohort, but unfortunately we don't have that data set yet. But yeah, I could raise it up in the lab.
Yeah. There are any other questions, Clive? Once again, thank you, Cecilia.
Hello, everyone. My name is Andrew Lawson, and I'm a postdoctoral fellow in the laboratory of Nico Martin-Carena, where I previously did my PhD. Today I'm going to talk about a recent focus in the cancer aging and somatic mutation program that's also been a major body of work that's in this lab, our lab, for the past 2 years. I'm sure many of you are aware, the cells in our body are not genetically identical to each other. From the moment the fertilized egg first divides, cells will accumulate mutations which are private to that cell and its descendants. By the time we reach middle age, most of our cells will have on the order of about 1,000 such mutations. These mutations accumulate as a result of intrinsic infidelity in our DNA replication repair machinery, but it can also be exacerbated by exposures to harmful substances in our environments, such as tobacco smoke and ultraviolet light. Now, our cells are remarkably able to tolerate many of these mutations without much of an impact. However, occasionally a mutation will arise that causes a cell to divide faster or survive longer than it otherwise would. These mutations are known as drive mutations, and when a cell accumulates on the order of 5 to 10 such drive mutations, they may become a cancerous cell. As you may know, we sequenced first cancer genomes back in 2008, and there's been a lot of work in the field in understanding how cancer cells are aberrant and different to normal cells. But in recent years, the department has actually been focusing on this earlier stage of the process. How do cells change their behavior when they acquire that first drive mutation? Now, you may think that studying the genomes of cancer cells should be harder than normal tissues because of how aberrant and unusual they are. They have different numbers of each chromosome. They have rearrangements. They have a large number of different types of mutation. But actually, detecting somatic mutations in normal tissues is more challenging. The reason for this is that in a cancer biopsy, many of the cells will share a mutation across all of them. These are called clonal mutations. And we can distinguish the cancer from its surrounding normal cells histologically. So we can take a pure cancer population and then sequence it, and we will see the changes. By contrast, in a normal tissue biopsy, the mutations will only be in a small number of cells, and we don't know what the clone size is. And often the clone size, even if we do know it, is very small, and it's challenging to actually sequence it. One way in which we can solve this problem is by taking a very small region of a normal tissue and hope that the mutations are at a high enough proportion of cells that we're able to detect it above the sequencing error limit. And this was the approach that has been used by the department for a few years. This is work for my PhD, where I was looking at mutations in normal bladder urothelium, the lining of the bladder. I spent a long time using a laser capture microscope to cut out 3,000 such little microscopic biopsies, and then we sequenced them using a mixture of targeted sequencing, exome sequencing, and whole genome sequencing. And then at the end of this, we're able to map mutations that have occurred in that individual's lifetime onto these individual microbiopsies. And one of the things that came out that was pretty remarkable to us is if you look on the left, there's a biopsy from a 59-year-old male, and you can see that ARID1A is coming up multiple different times. So this individual has a strong preference for acquiring drive mutations in that particular gene. By contrast, on the right, there's a 53-year-old female, and they have a strong preference for acquiring mutations in a different gene, KDM6A, which happens to be on the X chromosome. But one of the problems for this study was we only were able to look at around 15 individuals. Generating this data was very laborious, and it's hard to scale this up to large human cohorts. And so now, today, I'm going to talk about the progress we've made recently, which allows us to now unlock the world of somatic mutations in much larger cohorts. And the secret to this has been using a different form of sequencing. We now use what is called error-corrected sequencing. The version that we use here is called NanoSeq. And the principle of this is that if you take DNA and you fragment it, you can use the consensus of sequencing reads derived from both the top and the bottom strand of the original DNA molecule to greatly reduce the error rate of sequencing, because you're needing an error to have occurred independently in the two different strands to be read as a true variant call. This isn't a new idea. There are various other groups that have developed duplex sequencing style techniques. However, often in the library prep, the assumption that you have independence of mutations or damage between the two different strands is violated because you have some steps which copy information from one strand to the other. So we have generated a modified library preparation protocol which ensures that you don't have copying of information from one strand to the other. And so we now have a error rate on the order of 10 to minus 9, whereas standard sequencing has an error rate on the order of 10 to the minus 3, 10 to the minus 2. And so just to demonstrate this work, this is the results of my PhD project. As I said, 3 years of work to understand what's going in normal blood urothelium. And this is the result from a week's work with this new protocol. And you can see that we have detected more mutations in using this in this week of work than what took me 3 years before. And so we're now in a position that we can actually scale this up to thousands, tens of thousands of donors. And so we have applied this new technique to a collaboration with Twins UK, which would be fantastic partners, a very large collection of twins in UK. We've obtained prospectively 1,042 buccal swabs from them, and we've also had access to 371 archival blood samples. Here are some breakdown of the cohort, mixture of non-identical and identical twins, and then some twins where we only have one twin without a pair because the properties of that individual were interesting in their own right. We have a mixture of smokers and non-smokers. The cohort is biased towards women because the composition of the participants in Twins UK is also biased towards women. And we have some individuals that have histories of different types of cancer because one of the fundamental questions we're wanting to address is whether the somatic mutation landscape in an individual is predicting whether or not that individual may go on to develop cancer. So to start with, we're going to show what are the mutations that we detected in the blood. It's been known for about a decade that you can have this process known as clonal hematopoiesis, where a mutation occurs in a hematopoietic stem cell and it causes its descendants to take over the blood, reaching very high VAFs. And so for people familiar with this process, many of these genes will be familiar. This is something that's not unknown, but something I want to emphasize is that the proportion of cells that contain a mutation that you detect in normal sequencing will only be above that sequencing error limit. So it needs to be above 1% of cells, whereas we're detecting things down to VAFs of 1 in 10,000 cells, and we're really only limited by how deeply we sequence. So to put this in context, there was a paper study in Nature Genetics from a couple of years ago looking at UK Biobank. They sequenced 200,453 blood samples using standard sequencing, and so they were only able to detect mutations in those top two categories. We have sequenced only 371 blood samples using our new technique. We didn't detect quite as many drive mutations as them, but we have 25% as many drive mutations in a 500 times smaller cohort. Moving on from blood to oral mucosa, the buccal swabs. It's entirely unknown before this what the drive landscape was going to be in the lining of the mouth. And so as you can see, we have a large number of genes. Some of these are very similar to what we see in squamous cell carcinomas in the skin and what we see in the esophagus. And so some of them are not a surprise, but this is the richest description of a driver landscape in a normal tissue discovered to date. And what's truly remarkable is the number of mutations that we're seeing per gene. Again, to put this in context, this is showing TP53. This is one of the most common tumor suppressors genes, which controls whether or not a cell is likely to develop cancer across a wide range of tissues. We have detected a total of 8,114 mutations in TP53 in the buccal swab cohort alone. This is showing, for comparison, data from the Catalogue of Somatic Mutations in Cancer, a fantastic project that's been carried out in Sanger. It's celebrating its 20th anniversary this year. But you can see that we have detected more mutations in TP53 from our buccal swab alone than all of the data generated in squamous cell carcinomas to date. And that's because when you sequence a squamous cell carcinoma, you're likely to only detect a single mutation in that gene if it's a clonal mutation, if it's a clonal driver, whereas we're detecting tens to hundreds to thousands of very microscopic small clones simultaneously all at once. And then, so applying this approach across a number of different driver genes, simultaneously, we have a gene panel which is 239 genes, and we can apply X and Y, although that gets quite expensive. We now see a very rich driver landscape across these different genes. So for Notch1, we see this pattern of missense mutations in a certain region with nonsense and indels and central splice-like mutations spread throughout it. By contrast, in PPM1D, at the very end of the gene, we see a clustering of truncating mutations, nonsense mutations, and indels. For a different gene, RAC1, we see the pattern of mutations which are missense mutations, which are spread throughout the body of the gene in 1D space, but when you actually map them onto the 3D structure of the protein, they very nicely cluster around the GTPase binding domain. And then finally, there is an example of a gene called TP63, where almost all the mutations that we see that are selected are an essential splice site mutation at a very particular exon, but we see a range of those depending on which side of the exon interim boundary we're talking. So that's shown the power of being able to look at a large number of mutations from a saturation mutagenesis style approach. But the really cool thing about the work we're doing is wanting to get at the association between these mutation landscapes and risk factors for cancer, so in this case, oral cancer. And when we look at the pattern mutations, we split into two different signatures. There's the top one and bottom one, and we are now able to look at each of these different observable factors in our data and correlate it with known factors of the individual. So as we age, unsurprisingly, we see more mutations in our common driver genes, we see more total mutations, and we see you specifically accumulate this type of mutation pattern. Whereas if you drink heavily, that is causing almost all the mutations in this type of mutation pattern. When you smoke, it increases a mixture of both of these types of mutation pattern, but it also, on top of the increase in mutation burden, provides an environment where Notch1 mutations are selected more frequently. And you can see some unexpected results. So, for example, in type 2 diabetes, in the lining of your mouth, you're having a selection for more mutations in a particular gene called PPM1D. And if you have poor oral health, you have more mutations accumulating and more mutations in the gene called CHEK2. This is still a small proof of principle in terms of the scale of what you ultimately want to achieve. But the other question that we can get at this is whether or not there is the basis of the somatic mutation landscape is driven by environment, such as some of the things I've mentioned, or whether it's heritable. So in our cohort, if we look at height and weight, and we say how similar is the height and weight between two unrelated individuals, between non-identical twins and identical twins, unsurprisingly, we see that height and weight are very heritable. If we look at the two different types of mutational patterns, we see that for signature A, the pattern that is associated with increase with age, there is a trend for it to be more similar in identical twins and non-identical twins, suggesting that there may be a heritable component, whereas we don't see that pattern for the signature that we said was associated with smoking amount, sort of smoking and drinking amount. And then if we look at the fraction of mutations in Notch1 or TP3, two of our most common drivers, there are very clear hints at heritability, suggesting our germline is altering our frequency to have mutations in our somatic mutation landscape. So, final thoughts. This is introducing a new paradigm of how we might want to consider epidemiological studies. Traditionally, we have been able to measure risk factors in our lifestyle and then predict cancer risk indirectly from them. But we're hoping that by being able to directly detect what is going on in the clonal landscape, we can actually be able to measure those without necessarily knowing complete information about the risk, about the exposures. And ideally, although we're quite far from this, the dream would be if we could actually modulate the clonal landscape such that we select away from clones which go on to develop cancer. If you think of it as the equivalent of a statin for cancer in the way that statins reduce the risk of cardiovascular disease. The second benefit of this approach is that we've been able to interrogate many genes almost to saturation to be able to identify which mutations are likely to be driver mutations versus pathogen mutations. And so far, I focus almost exclusively talking about the risk of developing cancer, but now we're able to interrogate somatic mutations in a large number of normal tissues. There's a question of what role do somatic mutations play in other age-related pathologies. And finally, this is the most important slide of my talk. This wouldn't be possible without the work of many, many people. I'm very grateful to Inigo in particular for his supervision and Fedi for co-leading, but this has only been able due to research and development, pipelines, CHASM IT, and the CHASM Support Lab all helping support the effort. And then, of course, Twins UK have been fantastic collaborators with very rich and informative metadata. So thank you very much for your attention.
Thank you, Andrew. That's fascinating. Questions to Andrew? Caroline?
Hi, Caroline Relton. As a molecular epidemiologist, I think this area is hugely exciting. I've got lots of questions, but just to pin it down to one, have you looked at the relationship between somatic mutation frequency and DNA methylation or epigenetic factors?
So we currently have ongoing work for the department, which is directly addressing that question. At the moment, our ability to infer methylation changes and somatic mutation changes being correlated is limited to looking at clonal samples. So we're not using targeted analysis, we're using the laser capture microdissection approach that I mentioned before. It is in theory possible to use a similar strategy that we use in NanoSeq to actually get single molecule methylation change calling. But the problem is that if you wanted to correlate it with the somatic mutation changes, you want it to be constrained to individual cells, but at the moment we're looking at populations of cells. But yes, we are doing work.
Dan?
Dan Mahoney. When you look at sort of same individual, a gene gets hit in different places, how do you know that's due to, or how can you determine whether that's due to a convergent evolutionary process due to some similar selective pressure, or is actually due to a secondary structure of the DNA that's related more to the mechanism that's causing the mutation?
So half of that is based on the consequence of the mutation. So we use the NDS ratio of non-synonymous mutations. And also, if we look at the distribution of mutations in exonic versus intronic, we see very clear signal of selection rather than just mutagenic processes. That there are examples of signal we are seeing in the data which looks like it is just by mutagenesis. So we see this increased frequency of mutations in promoters, but that likely is solely a mutagenic process, whereas we can say for other regions of the gene that are being hit, it is very clearly likely to be selection.
Thank you. I'm a PhD student with Mark Blackster and Mara Lonizak in the Tree of Life department, and I'm pleased to kind of pepper this with some bits of the Tree of Life today. So I'm going to be telling you about painted ladies, butterflies like this that hopefully soon will be flying on campus, and painted chromosomes, which will make sense hopefully by the end of this talk. So I'm really interested in the evolution of chromosome structure, and that's because chromosomes are a key component of how our genomes are organized, and they also vary widely between species. So for example, in the Indian muntjac, they have just 7 chromosomes, while in the honeybee, there's 32. Hermit crabs have loads more, they've got 104, and in humans, we have the 23. And these changes in chromosome number come about because of large-scale rearrangements. So, for example, you can get fusion events, which is where 2 chromosomes come together to form the single larger one, or fissions where you get a break leading to 2 chromosomes. And these types of changes are really fundamentally important because they've been linked to biological processes like speciation. So, for example, in the North American spiny lizards, an increased rate of fusions has been linked to an increased diversification rate, so a generator of biodiversity. And also that implicated in adaptation. So, for example, in the free-spine sticklebacks, there was a fusion event between the sex chromosome and the other chromosome that brought together genes that are important in enabling them to adapt from a marine environment into a freshwater environment. So they're really fundamental processes, and yet there's a lot that we don't know about them. And so I'm really interested in Lepidoptera, which is the order of insects that includes butterflies and moths, and that's 10% of all described species. And so they're a hugely ecologically important group as well as being interesting from a chromosomal perspective. And that's because they show a huge range in chromosome number. They range from 5 chromosomes, like in Hyperphysphere on the left-hand side, all the way up to 223 in Polyomatus atlantica, which is just outrageous. So they have this huge variety, but if you're looking at this graph closely, what you'll notice is that actually this is kind of pockets of diversity that have these widely divergent numbers. And actually most species have a stable number of around 31 chromosomes, like what you see in Catocala fraxini. And so people have tried to think about this in the past, and one of the things that's kind of odd about Lepidoptera is that they fundamentally have a different kind of chromosome structure. So what we have in humans, and the same as in flies, like up here, is so-called monocentric chromosomes, where you have a single centromeric point where all of your machinery during mitosis and meiosis binds to and pulls your chromosomes apart. But in butterflies and moths, they do it differently. They have this activity along their entire length. And so what that's thought to mean is that if you get a fusion, you don't have two kind of point things that are antagonizing each other. And so that's can be resolved. And equally, if you have a fission event, those two new chromosomes can still successfully be retained in the daughter cells. So, well, this kind of doesn't completely solve it though, right? Because we still only have pockets of this diversity. And so basically we need more data and we need to understand this more. And this is just one example of a group where we don't know nearly enough about how chromosomes evolve. And in large part, that's because understanding chromosomal evolution requires chromosome-level genomes. And that's something that hasn't been made possible until relatively recently. And luckily, we're now in an advent of time where we're increasingly getting chromosome-level genomes across the tree of life. And not only across, but also at a depth that means you've got pockets of diversity with large numbers of genome sequenced with relatives. And so you can start to conduct these kind of analyses. So, for example, here we've got the white admiral, and each chromosome has been sequenced and assembled into a continuous sequence. And yeah, this is possible because of projects like Darwin Tree of Life and more recently Psyche. So my PhD, one thing that I've been looking at is trying to understand how conserved is chromosome structure across Lepidoptera if you actually look at the level of the genome. Are any chromosomes more likely to be involved in change than others? And are some species really genuinely more likely to undergo these changes? And if so, why? And I've been really fortunate because at the start of my PhD, there were 214 chromosome-level genomes available for this group. And excitingly as well, covered the breadth of diversity. So 16 superfamilies, including some of the very early ones, which allows you to really get what happened at kind of the base of the group. And 2 superfamilies of Trichoptera, so Trichoptera the caddisflies, which are the sister group to Lepidoptera. Okay, I don't have time to tell you all about this today, but excitingly a month ago this was published, so you can read all about it in the evening if you're really interested. But I can kind of quickly whiz you through some of the results. So what we first did was to understand what did the chromosome structure of that first butterfly or moth look like, to then use that as a framework of understanding how their chromosomes have evolved since. So we took the genes from all of these assemblies, we inferred a phylogeny, so the relationships between species, and then we walked up that tree and inferred what chromosomes were present at each set of ancestors, all the way to the base of the tree, which gave us 32 ancestral groups. So 32 chromosomes in that butterfly or moth around 250 million years ago. And we called these Merian elements, and they can be thought of as a framework similar to kind of Miller elements in Drosophila, if you've been taught traditional genetics. And we called them Merian elements after a lady called Maria Sabella Merian. He was an entomologist in the 17th century, predates people like Darwin, went around the Suriname with her kids, and described insect life cycles for the first time, as well as doing gorgeous botanical illustrations. So we thought naming them after her would be fitting. So we can use this as a framework and say, how have Lepidopteran chromosomes changed since the very first start of them? And what's really surprising is that 50% of species that we looked at haven't undergone any change, so not a single fusion or fission, despite that huge amount of evolutionary time. And the species that have undergone it, so those are the ones in orange on the tree, they've mainly undergone fusions, so we found around 200 fusions and very few fissions, so a clear bias towards one mechanism over the other. And what's really fun and quite unique about this group is that not only do they show this strong kind of constraint of chromosome structures, but also the genes even within them have kept an order. So, for example, we can look at the large yellow underwing and compare it to a butterfly, and what this straight line is telling you is that these genes have kind of stayed paired next to each other the whole way. And you can look at anywhere up the tree and you get broadly the same patterns. Take a really early diverging moth, and again, those genes have stayed in the same order, which is quite interesting. And so what you see when you get these fusions, to kind of give you a look at it, is these very blocky fusions. So, for example, this is the white ermine. Each of the rectangles you're seeing is a chromosome of the ermine moth, and the lines in it are the positions of the genes. And those lines are grey if they'd stayed on the same chromosome or Merian element since the last common ancestor, or a colour if they've moved. And so you can see here there's been a single fusion, and that's of the Z, the sex chromosome, to an autosome. And again, they've kind of kept in their own bits, so they've not mixed in between. But there are some exceptions, which I guess the very first graph I gave to you alluded to. And so what we found is that out of those 214 species, there were 14 that did something completely different, they've kind of thrown the Merian rulebook out the window and completely reorganized their genomes. And based on kind of where they appear in the tree, it seems that this is 8 independent instances of kind of genomic rearrangement. And so, for example, Melanea have done this, as well as Brentfest and Lysandra. And so you can look at these independent instances and start to understand, how does this happen? Why do some groups do this? And what is the way in which they go about it? And 2 clear patterns emerged. The first you can see from the blue butterflies. So we have the chalky blue Lysandra corydon fly around here. And what they've done is, again, each rectangle is a chromosome. As at the base of this genus, Lysandra, molecular scissors have become active, creating these fission events. And so they've rapidly cut up their chromosomes, resulting in 45 in Lysandra bilagus and 19 corydon. But just cutting, you'll notice. Which is completely different to what happens in the cabbage whites, where instead at the base of Pierini, some kind of molecular scissors became active, cutting up the chromosomes, but also a glue became active, sticking them together. So you kind of get this cut, stick, cut, stick, cut, stick, resulting in mosaics of Merian elements. So you have all these tiny blocks of conserved genes representing like complete scrambling. And what I think is particularly fun is because this process has been ongoing despite species diversification, is you get very different sets of chromosomes, but that can be disguised by a common chromosome number. So, for example, in Neporia, they've got 25 chromosomes, and Pieris also has 25. So if you were just to do traditional cytology and counted them, you'd say nothing's happened. But actually, they're completely different. They're separated by huge numbers of structural rearrangements. Okay. And yeah, we can start to unpick this, and we find examples where first kind of fissions have started to occur, and then the glues become active and start to unpick these 2 processes. So going back to the kind of the majority of Lepidopterans, what they do, what's powerful about having a large number of genomes is we can start to really look at the biases in how these things happen. So taking the 120 fusion events, we can ask, are some chromosomes more likely to fuse than others? And what we see is that length, chromosome length, is the key predictor. So here, each Merian element is a dot. The average length of it is on the x-axis, number of fusions on the y-axis. And you see the small chromosomes are much more sticky. They're fusing more often. And the exception is the sex chromosome, which despite being really long, is always fusing to those small ones as well. And so we can ask why. Why are the small chromosomes acting so different? And we can see that they are fundamentally different in a lot of ways. So, for example, their sequence content is different. They have a higher amount of GC rather than AT, indicating higher recombination rates. They also have more repetitive element content. And they also, I don't have time to show you, but different kinds of genes as well, more multi-copy and dynamically evolving genes. And we can ask if this kind of stays true, even in these really complex, highly rearranged species, are they still abide in this fundamental rule of length? And the answer is yes. So even though they're highly rearranged, they still show this negative correlation with length. So just to conclude, I think this kind of asks as many questions as it's answering. And so going forward, we're really interested in better understanding what are these molecular processes that are driving this high rate of fusion and fission. When we have all these independent instances, can we start to unpick whether the same sites are used, and if that tells us something about the biology of what genes have to stay together versus just tend to be? And really, why are some groups having this high rate of rearrangement, and what links does this have to speciation and the processes that generate biodiversity more broadly? So with that, I'd like to say thank you to everyone that helped with this study, especially my supervisors, Mark and Mara, have been extremely supportive. And none of this work would have been possible if it wasn't for the Darwin Tree of Life. And I think I'm very fortunate as an early career scientist in kind of an age where these genomes are being generated and are able really to do different scale of data sets than used to be possible. So thank you for listening.
So I'm Ewan Burney. I'm an observer on the board. I'm from EBI. That's a wonderful piece of work. You kind of hinted at that, what is the driver for the rearrangement? So I was wondering whether it was kind of an internal process, these salad moments, the times when they kind of blow up and they put themselves back together again. Have you tested whether there was kind of rapid evolution or some ecosystem change, or was it perhaps something more internal, some repeats gone absolutely crazy and there was like a repeat that you can see afterwards that you can't see beforehand or something like that?
Yeah, it's a good question. It keeps me awake at night. I think it kind of goes back to someone said earlier about kind of a chicken and egg situation, things like repeats. It's really hard to disentangle. Was there a repeat or proliferation because you've had these rearrangements, or did they drive it? And that's something people talk about a lot. And I think the only way to really get at it is look at very close relatives or even rearrangements that haven't fixed yet. So they're within populations, which is kind of what I'm trying to do at the moment. So this is great as an overview, but now I'm focusing on those species that are particularly interesting. And yeah, there's a lot of work going on in the tree of life, such as by Joana, looking at the link between these rearrangements and diversification. So that's...
And if I can just add, where did the W go?
So the W, so in LEPs, the Z is kind of the equivalent of our X, and the W is the equivalent of our Y in humans. And the W doesn't recombine ever, so it degenerates very quickly. It doesn't have gene coding sequence on it, really. And so you see lots of dropouts of the W where they're completely lost because there's no disadvantage. And they don't get included in analyses like this because it's based on genes. But yeah, W is also interesting.
I think you got one more question.
I actually did read the paper. It was an excellent piece of work. So well done. Question I have is, if you actually look, and I must confess, I'm fairly ignorant of butterflies, but if you look at the patterning of those 216 organisms, and yet you've got all these chromosomal variations, they all kind of, from a phenotypic perspective, they all seem to maintain sort of the same sort of wing structure and segment structure. Is that what one would expect if you start moving all these bits of genomes around?
Yeah, it's kind of hard. So whatever you think about, you need to justify it for why, they're so successful normally keeping them intact versus when they become highly rearranged. Yeah, it's a really good question. I think, there's some suggestions that, for example, the fusions to the Z, it can allow you to have a higher evolutionary rate because of the whole homozygosity. And so there you could get more rapid evolution, allowing you to, diversify wing patterns more. But the ground plan, like you say, is very conserved. There aren't these massive variations. And I think that's also why stability is so interesting rather than the exceptions, is did they kind of converge upon this framework that just works and they've optimized the number of chromosomes with recombination rate, and that allows them to, get the combinations they need appropriately. But yeah, it's really hard to think about it. And I think these pockets where there's a high rearrangement rate will be really interesting in looking at this, because it does coincide with high speciation rate.
Daniel Malawsky. I'm a PhD student with Hilary Martin and the Human Genetics program here. I don't have really exotic species to tell you about or really new cutting-edge technologies, but hopefully this is still interesting. I'll be talking today about the influence of autozygosity on common disease risk across the phenotypic spectrum in humans. So autozygosity is the inheritance of identical DNA segments from a recent common ancestor. And autozygosity can be elevated in the genome mostly due to two processes. One is endogamy, which is mating within restricted groups, such as the Finns. They're a well-known endogamous population. And consanguinity, which is mating between relatives, such as first-couple marriages. And both of these practices increase the probability of inheriting autozygous DNA segments in your genome. And the map here, for example, shows you the rates of first-cousin marriages or consanguinity across the world, where you can see that it's highly variable across the world, with some of the highest rates in the Middle East and North Africa. So more specifically, consanguinity increases both the number and also the length of runs of homozygosity in the genome, or segments of DNA that are identically inherited from each parent. And this the fraction of the genome that's in these runs of homozygosity has a very specific expected distribution for different degrees of consanguinity. So, for example, if your parents are siblings, you would expect 25% of your genome to be homozygous. If your parents are first cousins, you would expect 6.25% of your genome to be in these runs of homozygosity, and so on, as this schematic tries to demonstrate here. So in my research, I used 3 cohorts, or at least I'll discuss part of the research that I used 3 cohorts here. There's a bit of background. Genes and health is probably the least well-known one. It's a cohort of about 50,000 British South Asians, mostly collected at the time of this research from East London, with 31,000 British Bangladeshis and 19,000 Slovenians. We also use UK Biobank, the very famous cohort of around 400,000 British European ancestry individuals that we ended up using in our research, and then 10,000 South Asian ancestry individuals from this cohort. And after a really painful data sharing agreement process, I also am able to show you some results from 23andMe using about 500,000 individuals with siblings in this data set. And notably in Genes and Health and UK Biobank, we have linked electronic health records to all these individuals. But in 23andMe, we have only self-reported phenotype data. So first, we sought to characterize consanguinity in Genes and Health in UK Biobank. So in my master's in the group, I did in my master's here and my PhD as well, we were interested in developing a method to classify an individual, the parental relatedness, using only their genetic data. You can imagine many times this information on how related one's parents are is either not asked of the research participant, or it's frequently misreported, and also very frequently they may not know as well. In my master's, I developed this algorithm that allows one to classify an individual's parental relatedness using their genetic data, and it can classify you as to whether your parents are most likely siblings or avuncular or first cousins, which is this one category that I'll be discussing at length later in the presentation, this category of individuals with parents that are first cousins or closer. But it can also infer if you have parents that are first cousins once removed or second cousins or unrelated, which for the purpose of this presentation is parents that are further than second cousins. So when applying this algorithm in UK Biobank and Genes and Health, we find that about 2% of European ancestry individuals in UK Biobank are inferred to have related parents of any degree. And most of these individuals were inferred to have parents that are second cousin. But in the UK Biobank South Asians, 29% of individuals were found to have parents that are related. And this fraction was, again, higher in the Genes and Health cohort, which is composed mostly of British Pakistanis and Bangladeshis. But then when we looked at the British Bangladeshis versus the British Pakistanis in the Genes and Health cohort, which they reported their country of origin, we see that most of the high rate of consanguinity in Genes and Health is mostly driven by Genes and Health British Pakistanis, with over 25% of individuals being inferred to have parents that are first cousins or closer. So next, we wanted to look at phenotypic associations or trait associations with autozygosity. So it's well known that increased autozygosity is associated with traits across organisms, typically leading to worse health outcomes. In this example, you can see that moving from left to right, you have more inbred maize plants, and you can see a decrease in stature, which is observed across organisms, across several organisms, a decrease in stature with increased autozygosity in humans as well. But these associations are difficult to assess in humans because consanguinity is correlated with several social factors, including religiosity and the degree of educational attainment. So as an example of how these social factors can confound this research, in Abdullawi et al., they found that actually there's a protective effect of increased autozygosity on depression. So having more autozygosity in your genome decreases your chance of depression. But once they controlled for religiosity in this Dutch cohort where they did this research, that association disappeared, suggesting that just more religious individuals were more likely to have consanguinity in that population, just reported less depression. So it was mostly a social artifact that was observed in this association. With this caveat, that hasn't stopped people from looking at the associations between autozygosity and the genome and phenotypes. So in the largest such study, Clark et al. Conducted a meta-analysis of 119 cohorts to look at associations between autozygosity and the genome and 100 traits, and they found 32 experimental-wide significant associations. And these range from very different phenotypes, including height, as I previously mentioned, as well as various traits related to cognitive performance, reproduction, and behavior. But there's evidence in the paper and elsewhere that several of these associations may be driven by actually confounding rather than a true biological effect. So we were really interested in trying to tease this apart further. And the key observation that we made is that individuals with first cousin parental relatedness, so you're controlling for how related an individual's parents are, these individuals will still have very substantial natural variation in F row due to random inheritance of chromosomes and genetic combination. So in this little schematic here, you can see two individuals who have parents that are first cousins, and you can see that the individual on the left, due to the random inheritance of chromosomes and recombination, inherited a pretty short autozygous segment in DNA, but that the other individual who has parents that are first cousins was slightly more unlucky, and he or they inherited a much longer autozygous segment. But we have this algorithm that we previously developed that actually told us how related each individual's parents were. So what you can see here in the histogram there is the distribution of the fraction of the genome and runs of homozygosity when subsetting to individuals inferred to have first cousin parents. And you can appreciate that it's quite substantial variation, even though it's centered on 6.25%, as we would expect. So we considered in our analyses where we try to associate the degree of autozygosity or F row in the genome and traits to subsets of individuals. One is just including everyone in the cohorts, or the full cohort analysis, and one where we subset to only individuals who have parents inferred to be first cousins or closer, which in practice was mostly just individuals with parents inferred to be first cousins, because the other degrees of relatedness are very uncommonly observed in these cohorts. And I'll call this the highly consanguineous cohort. So the first thing that we did is we assessed associations between the fraction of the genome and runs of homozygosity, or F row, and various confounders that we were worried might be driving the associations that we might observe later. So we considered the association between F row and several different confounders here in UK Biobank European ancestry individuals. And we can see that F row is very strongly associated with religiosity, which in UK Biobank is someone says that they go to religious service once a week. So that's the phenotype, more specifically, that we're looking at there. So individuals that have much more autozygosity in their genome are much more likely to be religious, much less likely to have ever drank alcohol, much less likely to have ever smoked, much more likely to live in an above median deprivation area, and much less likely to have attended university. But once we subset to only the highly consanguineous individuals, these associations practically all fully attenuate. And of course, the highly consanguineous cohort is a much smaller cohort, so you may be wondering how much of this is just driven by the fact the sample size is much smaller. But power calculations suggest that this attenuation would not be expected just due to the decrease in sample size. And we conducted this analysis as well as the South Asian ancestry individuals in UK Biobank, and very similar results hold. So this made us feel pretty confident that this approach is doing an okay job at controlling for confounding. So next we were interested in the association between autozygosity and common diseases across the phenotypic spectrum. So we considered specifically 61 common disorders that were coded in electronic health records and Genes of Health in UK Biobank, and we meta-analyzed results from these two cohorts. And we found that F row is significantly associated with 12 diseases in the highly consanguineous cohort, with some of these including type 2 diabetes, asthma, as well as other anxiety. These are electronic health record codes, but some psychiatric disorders as well. And then in the analysis where we include everyone, so the full cohort analysis, we actually detected 31 significant associations. But interestingly, there was not a perfect overlap. So there were actually the psychiatric disorders we only detected as significantly associated with F row in the highly consanguineous cohort, and not in the full cohort. And there are two potential explanations for why this may be. One is that individuals that have parents that are more related actually have a protective environment somehow with respect to these two disorders. The other could be that consanguineous individuals are less likely to seek medical attention for these psychiatric disorders. And so evidence that I didn't have time to include in this slide deck right here suggests that the second hypothesis is more likely what is going on. So next we wanted to replicate these results to, again, really try to make sure that what we're saying is actually real and not just confounded. So the gold standard method in human genetics, at least, is to try to conduct within sibling analysis in order to determine causality. So this is a fully orthogonal approach that exploits the fact that within a family, we expect to see natural variation in F row as well. So in this case, in the full schematic, you can see two individuals and their siblings, and one just happened by chance to inherit a really long autozygous segment, and the other inherited a rather short one. Of course, they don't, unless they did 23andMe or got their genome sequence, they're not going to know this. So they're effectively blinded to the treatment. And FRO in this case is not going to correlate with environmental and social factors. So the basic idea behind this analysis is the question, does a sibling with higher FRO have a higher chance of having the disease? Of course, it's fit mathematically in a precise way, but that's the basic question. Of course, there's a caveat, of course, that 23andMe only has a self-reported phenotype, so it wasn't a perfect one-to-one match with electronic health records. But nevertheless, we were able to identify 7 phenotypes in 23andMe that correspond to phenotypes that we found as significantly associated in the highly consanguineous cohort analysis. In 23andMe, about 20, it depends on the phenotype, but the sample size was about 20,000 to 270,000 sibling groups or cliques. And these cliques tend to contain, on average, 2.1 siblings. So most of the analysis was just done by individuals that only have one other sibling in the cohort. And we found that actually, at Bonferroni-corrected significance, so experiment-wide significance, we see replication of 2 of the phenotypes, namely type 2 diabetes and PTSD, and which is very interesting because, of course, we only saw that the psychiatric disorders were associated in the highly consanguineous analysis rather than the full cohort analysis, suggesting that the highly consanguineous cohort analysis is doing a better job at detecting true biological associations. And we found that every single phenotype that we were able to assess in 23andMe that was significant in the highly consanguineous cohort had a concordant direction of effect size, which is significant, which is more than would be expected by chance. On the other hand, we then considered phenotypes that were only significant in the full cohort analysis, and none of them were Bonferroni-corrected significant, and only 29% or 2 of them had a concordant direction of effect size, which made us pretty excited, thinking that we did a good job here at finding true biological associations. The last thing that we were quite interested in is that there was a very strong associations between autozygosity, elevated autozygosity, and type 2 diabetes, which is interesting because South Asian ancestry individuals in the UK have 3 to 5 times higher rates of type 2 diabetes than European ancestry individuals. And we were interested, and of course we were also observed, and it's well known that there is much higher rates of consanguinity in British South Asians relative to European ancestry British individuals. And we were interested what fraction of that increased incidence of type 2 diabetes might be contributed by the fact that there is increased consanguinity in these populations. And we were able to assess this using population attributable risk, which is a counterfactual parameter that one can calculate that essentially calculates what percent of cases would disappear if a risk factor was removed, roughly. And in this case, the risk factor would be consanguinity, so having related parents. And we were able to calculate this population attributable risk for type 2 diabetes in the different cohorts. And we found that in UK being European ancestry individuals, the contribution of consanguinity is quite negligible to the prevalence of type 2 diabetes in the population. But then in genes in the British Pakistani individuals from Genes and Health, we found that the population attributable risk is about 10%, so quite substantial. But of course, it's important to put these parameters all in context. So, for example, high BMI is a really well-known risk factor for type 2 diabetes, and it's been estimated in several different cohorts because this is a kind of cohort-specific parameter that one calculates, but in several cohorts collected in India, the Americas, and Rotterdam, the population attributable risk for high BMI on type 2 diabetes is between 40 to 60%, so much, much higher than that attributable to consanguinity, even in the British Pakistanis in Genes and Health. And this paper recently published last year, which was a great collaborative effort, and it was a lot of fun, but now I'm doing something very different with the remainder of my PhD. And these are just the conclusions. So, we found that there's these differential rates of consanguinity between different populations. And we found that this approach of subsetting to highly consanguineous individuals attenuates confounding in this kind of analysis, assessing associations between autozygosity and traits. we found that FRO is associated with several disorders, including type 2 diabetes and anxiety-related disorders such as PTSD, with pretty convincing replication and a within sibling analysis in 23andMe. And we found that there's a significant contribution of autozygosity to type 2 diabetes prevalence in British-style Beijing's. And with that, I would like to acknowledge, everyone in my group, especially Hilary Martin, who is my supervisor and really saw this all the way through, and Jared O'Connell at 23andMe, who was a really important link at 23andMe to make that portion of the analysis available to publication and, get it to where it is today. So, and I would like to thank everyone else on the slide and, of course, all the research participants across these 3 cohorts. So thank you.
Hi, everyone. I'm a group leader at the Tree of Life program. So the human genome has been absolutely transformative for understanding us humans and for helping us keep healthy and so on. And we've had some really nice talks by PhD students and postdocs really highlighting this single-cell sequencing or spatial transcriptomics of different tissues would not be possible if those reads couldn't be aligned to a reference genome. At the same time, like studying cancer and somatic mutations would not be possible if those reads couldn't be aligned to a reference genome. So the first step to do all these kinds of things is really to have a reference genome. Now, at the Tree of Life, we think that if we generate reference genomes, not just for humans, but for all kinds of species, we will really not just transform human health, but also planetary health and help ourselves in the meantime. So we think that reference genomes will have a global impact, affecting biomedicine, biomonitoring, sustainable fisheries, etc., etc. I'm not going to read all of them in the interest of time. But we really think that having the reference genome is really the base for what you need in order to do any kinds of genomic studies. And the Sanger Tree of Life is leading the world in biodiversity genomics. And we're doing it in a very open way, encouraging others to follow our example, making all the reference genomes openly available, and allowing all people doing all kinds of things listed here to use our reference genomes and transform their fields. So we do this in multiple large projects. The biggest that you may have heard of is the Darwin Tree of Life project, that's sequencing all 70,000 eukaryotic organisms in the British Isles, Britain and Ireland, from land and sea. And so that includes charismatic taxa like this orca here that was sadly stranded but gave us a tissue sample, or very important pollinators or the plant it sits on. And we don't just do this alone, we do it in collaboration with lots of institutions across the UK that provide samples that are then sequenced here at the Sanger Institute and transformed into reference genomes at a very high quality. And so we already have produced over 1,200 genomes that are openly accessible and available for everyone. You can see that we've covered already a large proportion of the Tree of Life. With a weird, you can see there, I can't really point, but with a weird overrepresentation of butterflies and moths, then you can see that this has been used very well by Charlotte. We have over 5,000 species in progress, and the number of reference genomes per month is just going up and up and up. It's really an amazing project. Just to make clear what these numbers mean, so across the globe, all the reference genomes that are at chromosome level quality, the Wellcome Sanger Institute has produced 36% of those genomes globally available. And if we just restrict it to the last year, it's 46% of the genomes. So we are really leading at the front of producing reference genomes at the scale that's unprecedented and not seen in any other country or project. I've introduced the Darwin Tree of Life project already, but that's not the only one. There's, for instance, also the Aquatic Symbiosis Genomics project, which is sequencing symbiotic systems from marine and fresh water, like, for instance, a coral, which is actually a combination of animals that are most closely related to jellyfish and algae that are photosynthetic. And so they are sequenced together. It's impossible to really disentangle those. they're sequenced together, and that requires new lab techniques to extract DNA from something like that. And then new bioinformatics pipelines to disentangle the genomes to then afterwards have really nice genomes of both the animal and the algae. And then recently we launched another project that Charlotte has already briefly mentioned, which is Project Psyche. This project is in collaboration with lots of different institutions across Europe. And here the aim is to sequence all 11,000 species of butterflies and moths. And you could see from Charlotte's talk what one can do with this kind of data. And now expanding it to 11,000 species will really allow us to gain a really good understanding of the evolution of butterflies and moths. But these really are kind of just an example of a taxa where we can find out lots of things that can then be generalized across all organisms. And of course, they also include important agricultural and close pests and are important pollinators and biodiversity indicators. So very important organisms. Now, how do we make such reference genomes? The first step is collecting the specimen. And that's not easy when you try to collect all taxa across the UK, for instance, or all butterflies and moths across Europe. So it really requires collaboration with lots of people who are experts in specific taxonomic groups that know where to find them and also how to identify them. And we do this all in a very standardized way. Then we fill out spreadsheets with all kinds of metadata, so we really know what's what when it all comes together in the thousands at the Sanger Institute. And then before we accept any specimens into the Sanger, a very rigorous system of compliance checks gets kickstarted with different teams with expertise in legal and ABS, research governance, etc. Check everything before we actually start sequencing. And so the sequencing of a reference genome requires two parts, and that's PacBio sequencing, long reads, which allow us to puzzle together DNA sequences. And so we can get a sequence that captures all this information. But then there's always these annoying parts which are highly repetitive and it's really hard to sequence through. So we also add in high-C sequencing, which allows us to stitch together these contigs into chromosome-level scaffolds. And then in addition, for each individual, we also do RNA sequencing, which allows us to then afterwards annotate the genome and shows us where the genes are. And so we get a very nice chromosome-level genome, and the genome annotation is done by EBI. When we sequence a genome like that, we actually don't just get one genome, even if it's something like a moth. We get the chromosomes of the moth itself, but then we also get its mitochondrial genome. And in this specific specimen, there were actually 3 different strains of Wolbachia that were also sequenced that we can also assemble. So we basically get 5 genomes, even though we only attempted to sequence the moth itself. We can really sequence gigantic genomes that also required a lot of method development. This mistletoe has a genome of 94 gig. So each of these chromosomes, each of these squares with the X's you can see there, are 3 times as big as the human genome. We can also sequence tiny, tiny, tiny specimens which don't have very much DNA in it and therefore really hard to sequence, like a nematode, a single nematode worm, I should say, where we can get a really nice genome out of that. Again, that required a lot of method development in the lab and bioinformatically. When we have the genome ready for release, we also publish a genome note, which is basically a small publication that introduces the genome, acknowledges the people who have contributed the specimen, and also produces or also shows some automatically produced graphs like these ones that highlight the quality of the genome. Then we have developed lots of different portals where people can see which genome has already been sequenced, either by us or even by other projects as well. Because we are leading many of the new projects starting, they all just use our databases and portals. And that's great because that really allows global coordination of sequencing projects to avoid that lots of projects all focus on the same species. And so this is Genomes on a Tree. That's the portal that allows the coordination of the sequencing. At the Tree of Life program, we are really world leading in the unprecedented scale, the number and quality of genomes produced, but also in open access sharing. Really sharing all the protocols that we use in the lab and bioinformatically, making the genomes immediately available openly for everybody to use. Coordinating sequencing efforts across the globe. We are also engaging strongly in training in biodiversity genomics and facilitating the startup of other projects, like, for instance, the African Biogenome Project, which aims to sequence with a focus on African species sequencing in Africa, or the European Reference Genome Atlas, or Atlas C, a French project focusing on marine organisms. Then there's also Genome in a Box, which is recently launched, where we finally have funding to kickstart this, which is basically allowing us to put everything that's required to generate reference genomes in a box, ship it to Guyana, for instance. That's going to be the first place. And then they can produce their own reference genomes at biodiversity hotspots, which don't have the sequencing facilities that we have here. So really helping others to follow our lead. In the same spirit, there's also the Biodiversity Genomics Conference, which is 5 days of talks across different time zones, and therefore at different times. Some of them are at night for us, but not at night in Australia. And so they're focused on different geographical regions. And as part of that, there's also the Biodiversity Genomics Academy, which last year hosted 40 sessions of short courses teaching people how to use bioinformatic tools to produce reference genome or to use them to answer their questions. And last year there were over 1,200 participants, and all the talks and tutorials are still on GitHub and YouTube and regularly watched and clicked at by lots and lots of people. So this was kind of like the giant projects, but we are 4 faculty, Mark, Mara, Kamil, and me, and we all have our own projects and interests that we use the reference genomes for to answer specific questions. Like me personally, I'm interested in why do some lineages diversify very rapidly and others do so at a much slower pace. And reference genomes really allow us to answer these kinds of questions. But because the reference genomes are openly accessible and available to everybody, lots of other people also use these reference genomes, and I just want to highlight 3. This is a biodiversity conservation project where sea eagles are reintroduced, and Cardiff University is studying with genomics if the sea eagles that are reintroduced to the UK are the right strains, if there's specific local adaptations that they're missing that are present in other countries that we should get eagles from to reintroduce them here. Or agricultural project, the Wellcome Institute is developing an early warning system for crop pathogens by sequencing the air. Or human health, it turns out that predicting if a variant is related to disease or is likely to be disease-related works quite well if you just look at are those gene sequences conserved across a large number of species, and if they are, then mutations are very likely to be disease-related. That's just to highlight 3, but I hope at the beginning I could show you kind of like how the impact can be very far-reaching and affecting lots of different areas. And if there's time, I'd be happy to answer questions.
So my name is Gosia Trynka. I'm the Science Director of Open Targets Associated Research Program, the Sang Institute, and I also run a research group in the Human Genetics Program. And so I'll give you today an overview of Open Targets, what it does. It's an academic industry partnership to accelerate drug discovery. So we're a partnership with the goal to transform drug discovery through systematic identification and prioritization of targets. And we're a partnership between the EBI, Sanger Institute, and currently 5 different pharma partners that include Genentech, GSK, MDS, Merck, Pfizer, and Sanofi. And the kind of the backbone to what we're doing is the fact, or something that really fed to the inception of Open Targets, was the fact that genetics increases the chances of success of drug discovery. And depending on the genetic evidence that feeds into this, the likelihood can be either something like 8 times greater success or 2 times greater success of getting a drug approved through clinical trials. And more recently, actually, when we in Open Targets had a look at the FDA drug approvals in the past decade, we identified that there is a support by human genetics evidence for 63% of all the targets. So clearly, being able to leverage genetic information better will accelerate drug discovery. So in Open Targets, we generate our own data and use publicly available data sets to find potential new drug targets. And we focus on this kind of 3 pillars on the data, knowledge, and decision making. So on the data side, we collect types of data that help us build relationships and confidence in between this target and disease links. So really building the evidence of a target in the context of a disease. On the knowledge side, then, we expand this information to provide more context of a biology for a given target. And that then enables us to build therapeutic hypotheses. And the final pillar of the data that we integrate is around decision-making when thinking about therapy. So whether a target is targetable, has it been, are there already clinically approved drugs for it, where in the clinical trials they are, why clinical trials fail when targeting certain targets, and so on. So collectively, this builds like a holistic picture that helps to envision safe and effective medicines to catalyze their progression. So our research program is realized really by a true collaboration and interaction closely between the EBI and Sanger, and that is reflected by some of our core teams that are situated either at EBI or at the Sanger. On the leadership side, I'm based at the Sanger Institute. David Halkop, who's the executive director, he's at EBI, and Ellie McDowell, who's the translational informatics director, is also based at EBI. Our core platform development team is all based at EBI. And then we have a genetic analysis team based at Sanger, as well as validation lab also based at Sanger. So as you can see, it truly is a very close collaboration between the 2 institutes here on campus. And in addition to core teams, we also very closely interact with all of the faculty based on campus through a range of different research projects that we fund. So we lead an integrated research program. That means we closely intertwine the data that we generate or that we ingest from publicly available resources with our informatics capabilities. And that is on the informatics side predominantly to build this target identification information and target prioritization. The genetic data is used to provide a genetic evidence with a clear focus. And then data integration and visualization are really important aspects of what we do. And that can be then browsed through the platform, which I'll come back to later. On the experimental side, we capitalize on the Sanger capabilities, and we leverage disease-relevant systems that are available at Sanger through the faculty in here. We look at, we use systematic perturbations in the context of specific biological questions, and we'll use a range of really multivariate readouts that are available at the Sanger. So 80% of our projects involve collaborations across academic teams, and all of our projects collaborate with industry teams. So that means that really rather sporadically we interact with one academic team only. Most of the time our projects bring multiple groups together. Of the 22 eligible faculty at the Sanger Institute, 12 participate in Open Targets projects, and by eligible, I mean programs that are most aligned with the Open Targets vision. So that includes human genetics, CellGen, and CHASM. So 50% of all those faculty in those programs actually collaborate with Open Targets. And 30 of our currently active 38 projects involve Sanger faculty. And 23 of these projects feature Sanger cross-program collaboration. So we not only have collaborations within the programs, like, for example, within human genetics, but we also actually have quite a lot of collaborations across institute between the different programs. And in addition to that, 8 projects also have collaborations with EMBL EBI, and 20 with additional external collaborators, often with clinical collaborators who work with us by providing different clinical samples. So our core platforms include the flagship Open Targets platform, which you can go to by typing this address. The goal of the platform is to prioritize target disease associations. And this is done really by systematically harmonizing all sorts of different publicly available data types. And this harmonization and integration is really very important part of what we do. And we use over 20 different sources of evidence to do this, to build this prioritization for the target disease associations. And we systematically prioritize target disease associations and provide the ability to filter and explore what are the different types of evidence behind those priority scores. Our second core platform is the Open Targets Genetics, which currently has its separate instance. I've lost the laser, but you can type that address and it will get you there. And that is only focused on the genetic evidence. And for that, we use a range of different data sets that are, again, publicly available or that come through internal efforts here through Sanger. Similarly to what Platform does, a very important part of this is harmonization and integration of large genetic and functional genomics data sets. Also, a very important component is the statistical analysis that the team carries out, including this own score called locus to gene score, which provides a quantitative score of how likely a gene is to be a causal gene for a disease. And our final core activity is the Validation Lab. This is an experimental platform. The goal here is to deliver systematic and independent target validation at medium throughput. The Validation Lab really is positioned to be directly in between the industry and academic teams. Some of the core responsibilities for the team is to assist in silico target triage for validation, and then they perform independent experimental validation of the results. And through these activities, they also expand on the biological context of a target through the validation activities. So, impact of our program. So since its inception, Open Targets has raised £114 million, and half of this has been awarded to Sanger through 72 out of 93 total of our projects awarded to date. So really half of the funds go to Sanger and half go to EBI. And we have published 70 research publications since the start of the program. And I'll walk you just through the two quick examples of some of the projects that we have run in the past as part of the program. One focusing on cancer and mapping cancer dependencies, and the other one, a work led actually in my team, looking at immune regulation I'll walk you through it. Project SCORE was one of the very first projects that we designed in Open Targets. This was a genome-wide CRISPR-Cas9 screen across 330 cancer cell lines, looking for what are the synthetic vulnerabilities of cancer dependencies. So all of the data became publicly available. And what's interesting here is really to look at the timeline of how we work with our industry partners and what is the benefit and impact to the industry partners. So the project was designed in 2015, and the first results started to emerge and be shared with partners about a year later. And then 2 years since the inception, the Werner helicase was discovered as the essentiality gene for multiple microsatellite unstable cell lines. And then in 2018, academic and industry team worked together on validating this result as a target for drug discovery. And so this project was published in 2019, so really 4 years since the inception. But from the very beginning, there was a very close interaction with industry partners. They had access to all the data, and they were able to benefit from that information as soon as it was emerging. The other project I wanted to highlight to you is a project that was run in my group. And in this project, we focused on the role of CD4 T cells and the activation on CD4 T cells and how this process is important for a range of different immune-related diseases. And we've generated a range of different data sets, one of which was first of its kind single-cell eQTL map of T cells as they progress through activation course from resting through different time points of activation. And we identify a number of different genes that were regulated by the same variants that predispose to the development of immune diseases. And this type of data set, although we didn't identify a single target, it actually had quite a substantial impact to our industry partners. As we heard back from them, the data was used and is being used internally for a target triage team, especially in the immunotherapies, has informed a number of different variant to gene to function efforts, and actually has been used to actively pursue over 10 target experimental design decisions. And beyond the academic institutes and the industry partners, Open Targets also has quite substantial impact on the community. So these are just some examples. Our numbers of users that engage with the platform increase from every year. And more importantly, we have become really like a gold standard for what a causal gene is. So, for example, we're routinely used to support hypotheses this is just an example of an extract from a manuscript where the authors really go back to Open Targets to use us as a ground truth. Because of our efforts in harmonizing the data and bringing it all together, the data sets that we publicly release are becoming very important for supporting machine learning because they are well structured, and also contribute to both commercial and public instances of knowledge graphs. And the final example is our code for the platform is publicly available. And last year, there was a kind of a spin-out of almost like a mirror of our platform from NIH CHOP Institute, where they developed their own instance of a platform specifically focusing on pediatric cancer. So really leveraging the open source code that we release and the similar framework to dedicate efforts toward something that we're not doing. And to close, Open Target's been around for 10 years, and in October we will be celebrating that we are 10 years old, so you may have received some invitations for those celebrations. Thank you very much.
You take them.